Dear learners, in my last lecture I discussed C4 pathway. C4 plants can avoid photorespiration and achieve higher water use efficiency, higher nitrogen use efficiency. In this lecture I am going to discuss another variant of C4 pathway that shares some similarities as well as some differences with the C4 pathway called as CAM pathway. C4 plants have two types of photosynthetic tissues, mesophyll and bundle sheath, whereas CAM plants have a temporal arrangement. C4 plants have a special arrangement of two types of photosynthetic tissues, whereas CAM plants have a temporal arrangement of inverted stomatal cycle. That is, they open their stomata during night time to allow entry of carbon dioxide and close their stomata during daytime to conserve water, because they are typically found in the more drier desert environments. So they have an inverted stomatal cycle. CAM is short for crystallization acid metabolism. Because this phenomenon was extensively studied in the family Crasulaceae. So this was named as Crasulaceae. And there is a story about acid metabolism, that a poor thirsty man who was lost in desert squeezed cactus plants to meet his water requirements. During daytime that liquid was tasteless or didn't taste good, whereas during night time it had a nice acidic taste. So by the end of this lecture I hope you will be able to understand why it tasted nice or acidic during night time. So although this phenomenon was extensively studied in the family Crasulaceae, but it is also found in some other families like Cactaceae and Euphorbiaceae. C4 plants have Kranz anatomy, that is a tightly fitted layer of cells around the vascular bundles called as bundle sheath cells, whereas CAM plants like pineapple, agave, cactus, they exhibit succulence. They don't have specialized Kranz anatomy, but they have thick fleshy leaves or stem in case of cactus, which have thick fleshy photosynthetic stems with large central vacuoles, large water-filled vacuoles. So CAM plants exhibit succulence, but it doesn't mean that all succulent plants are CAM. So let me now draw the CAM pathway, how they fix carbon dioxide during night time and how they keep their stomata closed during daytime. So first, what happens during night time? During night time, their stomates are open, so they allow the entry of carbon dioxide. And you know that when stomata are open, water can also leave the cells of plants. As in case of C4 plants, its first substrate or first acceptor molecule is phosphoenol pyruvate that prefers bicarbonate ions as for its carboxylation, and this enzyme, this step is catalyzed by peptase enzyme, that is PEP carboxylase, same as in C4 plants, and forming a 4-carbon acid, that is oxaloacetic acid. Let me write it full form here. This oxaloacetate is reduced to malate using malate dehydrogenase enzyme, and this malate is stored in plant vacuoles. So that is why those cactus plants, liquid of those cactus plants tasted acidic during night time. And the source of this first carbon dioxide acceptor molecule, phosphoenol pyruvate, is the starch in chloroplast. This was made during daytime. This starch is converted into triose phosphate, which can form phosphoenol pyruvate. So in this way, carbon dioxide is fixed during night time. And what happens during daytime is the stomata are closed usually, so carbon dioxide remains out. It cannot enter the plant cells and water remains in, thus conserving water. This malate that was stored in vacuoles is converted to 3-carbon compound pyruvate. Malate undergoes a decarboxylation reaction, releasing carbon dioxide. So, as we discussed in C4 pathway, in the same way, this carbon dioxide can be fixed via Calvin cycle or C3 cycle or photosynthetic carbon reduction cycle, forming starch. This pyruvate can be converted to triose phosphate, and this can also be converted to starch. So this starch is the source of energy during night time that is required for carbon dioxide. So this is the source of phosphoenol pyruvate via triose phosphate. So that is how can plants can fix carbon dioxide and do photosynthesis ultimately through C3 pathway. But in this way they achieve highest level of water use efficiency. So as I told you before that C4 is a special pump in which carbon dioxide is concentrated through mesophyll and bundle sheath cells, whereas is a temporal pump in which stomata remains closed during daytime and open during night time. But there are some additional energy cost involved in the C4 pathway for the generation of phosphoenol pyruvate or for accumulation of malate. For 1 molecule of carbon dioxide fixed in C3 plants, we need 3 ATP and 2 NADH. In C4, we need 5 ATP and 2 NADH, whereas in CAM plants, we need 6.5 ATP and 2 NADH. So there is additional cost for this. And this is different from seafood plants also in the sense that PEP is derived from stored starch instead of the carbon intermediates as in C4 cycle. Canned plants can retain and de-assimilate respired carbon dioxide as well. So there is at night time there is less evaporative demand because for the same water content, the cooler it is, the higher is the relative humidity during night time. So in this way they can conserve their water. That is why these canned plants like pineapple, agave, cactus, they are found typically in desert environments. There is another question that which pathways came earlier, whether it was C4 or CAN pathways, and the answer to this is that CAN pathway is more primitive. It came earlier than C4 because this is also found in primitive ferns, whereas C4 pathway is only found in angiosperms. So it shows that this is the evidence that CAN pathway came earlier than C4 pathway. CAN plants are about 10% of total terrestrial plants, whereas C3 plants are only 3% of terrestrial plants. Some of the CAN plants are obligatory, like cactus plants, so they only have CAN pathway, and some are facultative, they can switch their mode. For example, ice plant is a C3 plant. It has a C3 mechanism. Its stomata remain open during the daytime and closed during the night time. But under stressful environments like low water availability or under high salt concentration, the ice plants can switch from C3 to CAN mode of carbon dioxide fixation. That is how CAN plants can fix carbon dioxide and also conserve water.